Okay, so thanks a lot for coming today. So on behalf of the same organizing committee, we wanted to wish you a happy new year. And just to let you know that we will announce next week the results of the call for the spring semester speakers. And with that, we will start with today's seminar. So as usual, people who are on Zoom, you can report any technical issue in the chat and ask your question in the Q&A tab. And we will allow you to speak at the end during the discussion session. And with that, I will leave it to Nicolas to introduce today's two speakers. Thanks.
Hello, everyone. I wish you all a happy new year. And it's my pleasure today to introduce, today it's a bit unusual for the same seminars because we have a kind of a joint presentation, although Lucile will be the only one talking here. So maybe first I introduce Sarah Benhaiem. She's working at the Leibniz Institute in Berlin, where she did her PhD. So she's working on hyena. She has a project in Tanzania where she's been working during a PhD, postdoc, and now she's a group leader at the Leibniz Institute. And she's working on behavioral ecology, population dynamics, and disease ecology. And Lucile Marescot here, she's working, she's doing models in population dynamics. She did a PhD at the CEF here in Montpellier. Then she did the first postdoc at UC Davis in the US, and then a postdoc at the Leibniz Institute. And she'll be talking about the project she was conducted there with Sarah today. And then she did a short postdoc here in Montpellier, and now she's a researcher at CBGP here in Montpellier, working in population dynamics of locusts. So she moved from large carnivores, kind of more iconic, to like insects. And with this, yeah, the floor is yours, Lucile.
So good morning, everybody. That's Marsha. Yeah. So it's a great pleasure for me to be here today and to give this talk. I'm going to present you an overview of a different study we conducted with CIRAD during our postdocs in 2017-18 in Berlin, in the Leibniz Institute, in the Zoo and Wildlife Research Institute. And I'm mostly going to be talking about, so I'm sorry if I'm a little bit lacking of energy. I'm recovering myself from COVID last week. And I'm going to be talking about disease transmission, funny. And a case study, so I will be talking about the impact of an epidemic of canine distemper virus among the spotted hyenas, among the spotted hyena population in the Serengeti in Tanzania. So the negative effects of pathogen transmission, of pathogens that are transmitted by social contact, so either through physical contact or social proximity, is today one of the major issues in the world economy and human health. So as you may know, a lot of zoonoses have emerged, and COVID-19, Ebola, HIV have caused already millions of deaths and a lot of economic damage. So it is really important to study the pathogen transmission in many species, and especially in social species, in order to really assess how the society can shape the disease transmission, the pathogen transmission, and the fitness cost of infection. In order to better to limit the demographic consequence of infectious disease in socially structured population, and of course prevent the spread and persistence of pathogen on the long term. So what is a socially structured species? So first, let's agree on the definition of a social species. So I'm going to use the definition of Wilson, that considered any species as a social when it lives in a stable group, in stable groups exhibiting reciprocal communication of a cooperative nature. And so life in groups, in social group, has been considered as one of the major transitions in evolution, and it presents a lot of advantage to individuals. So whether it is known as a great anti-predator adaptation, so by reducing the per capita predation rate. It can also increase the defense of resource and foraging ability of individual within the groups, and favor the selection, mate selection and breeding success. But these, of course, comes with some costs related to pathogen transmission, and due to a higher contact within the groups, we have higher exposure. Individuals have higher exposure to virus and pathogens, and higher risk to contract the disease. And of course, and also in groups, individuals are often related genetically and have very similar immunogenetics. So these costs, so to really understand better the demographic consequence of disease on the population level, we really need to first understand the links between sociality, health, and fitness of individual. So regarding the sociality, of course, we have a lot of different variation behind this concept. So you have different species that can live in groups of different group size, different type of interaction, and social hierarchy. So all those social organization can vary between different species, different behavior. And this variation and heterogeneity in social contact influence, of course, the health of individual and how it respond to pathogen exposure, and how it can have consequence in terms of fitness. So, for instance, you have a key trade-off between reproduction and immunity it can be mediated by social status, where low-ranking individuals have a higher stress and higher level of glucocorticoid, and they're going to be much more affected by pathogen exposure, and which in return is going to compromise their reproduction success. We can also acknowledge, of course, the feedback between health and sociality, because infected individual can also behave, once they're infected, they can behave differently or be treated differently within the groups. And you have also many studies, that have shown that, for instance, in bees, that division of labor and caste system could have evolved under the selection pressure of pathogen. So as you can see, all this is quite complex, and there is really a lot of evolutionary hypotheses that has been tested and that can still be tested. But today I'm mostly going to be focusing on the demographic response to outbreaks. So how pathogen affects the health of individual, and how this can be mediated by social traits within the population, and what are the fitness costs. And then how does it transfer to the population level in terms of the population dynamics and disease dynamics? So we will try to address two main questions, and what are the impact of pathogen on the health and fitness of individual at the individual level, and also how does this pathogen can impact the performance of the population at a population level, and how all this is mediated by social factors. And then the main other question we will address is how those social contracts within the group and between the groups can influence the transmission and persistence of pathogen. So to do so, we are going to use a very fascinating model. So for the host model, we have the spotted hyena, which is very interesting to study the pathogen in age and socially structured population, as the spotted hyena is a highly social carnivore that is a caste-selected and long-lived animal, since it has a really long lactation period and a very high extensive maternal care. The hyena lives in stable groups that are called clans, and they are structured in a linear hierarchy where all the females dominate the males, and the cubs are sheltered within communal dens. They also have those, describe those long distance commuting trips to foraging area and to follow migratory herds. So as I said, the females are dominant. So you have a linear hierarchy where the alpha females dominates all the other females, and then the males are at the bottom of the hierarchy. and also the offspring of females inherit the social status. So you can have very low cubs that are that have a higher social rank than a big adult male, for instance. And what else? It is also a female. It is also a philopatric animal, a female philopatric animal, where the females stay within the clan and the male emigrate and migrate to mate. And of course, there is also high competition among the siblings, among the cubs within the same litter, and sometimes some siblicide have been reported. So this is just a general overview of the biology and ecology of the hyena. And then in terms of the pathogen model, we have the canine. In all along this talk, I'm going to talk about the canine distemper virus, which is also quite interesting with a similar behavior. Well, I'm not going to make an analogy with COVID, but it's a RNA virus. It is a part of the morbillivirus family. Which the symptoms of the disease can be respiratory, gastrointestinal, and also affect the nervous system. It is also a multi-host virus affecting mostly carnivores, and some studies have also reported some zoonotic risk. It's spread through the air, so it's an airborne vector disease, but also through physical contact. It is highly contagious, and once you recover from it, it confers a lifelong immunity, like measles in children. So during this talk, I will be presenting the 3 different studies we conducted during our postdoc. The first one is about how to disentangle all the eco-epidemiological process in this socially structured species, and to determine the individual fitness cost of infection. Then we looked at how the resilience and the resistance of the population to disease outbreak. And in a third study, we looked at the disease dynamics and herd immunity, how it can be affected in clan-structured populations. So the first one is mostly at the individual level, at the population level, and at considering homogeneity between clans, and here looking more at between clans dynamics. So we used a long-term data set obtained from a long-term monitoring that is still going on. It started by 1987 by Marion East and Herbert Hofer, where they followed regularly individuals in 3 different clans, and in the Serengeti National Park. And we have this CDV, so the canine distemper virus project, where we have we could follow the health condition of each individual in terms of whether they're infected or recovered. And we have information about the demographic states and the social status of 25 female and 816 males during 20 years, so from 1990s to 2000. So we considered different social states where high-ranking females, so all the females in the linear hierarchy that are above the median, and low-ranking female that are all the ones that are below the linear hierarchy. And of course, it is quite dynamic system where the female and male, well, female can also change rankings, social rank. Then we have the demographic states where we followed the individual from their cub stage to the subadults, from the then breeders or non-breeders, and all along their life cycle. Now, regarding the transition between disease states and epidemiological states, we have gathered all the serological data with the CDV antibodies. These were collected when hyena were anesthetized in snare removals, and the PCR screening of tissues and feces when the hyenas were found dead or again anesthetized to remove the snares. The serological data allowed us to assign the recovered states of individual while the PCR screening was to identify the antigen, the CDV and the infected state, and also clinical signs were allowing us to, without doing PCR screening, to also assess whether an individual was infected from the symptoms. So we had blue, Sheen's eyes, respiratory disease, ataxia, and body faces. I can also have a respiratory problem, and nasal discharge. So we ended up with a capture-recapture data set of, so as I said, 20 years of monitoring, individual monitoring, where we can follow their health condition over time. And of course, it is always, we're not going to have information about serological and any information on disease states for all individuals. So we're going to have sometimes uncertainty about the disease state. Sorry. So this is just something to account for. So basically, we ended up with 22 possible states for the hyena population. So where we have the different demographic states, the social status, and all the transition that can occur between those states within one clan that is structured around a communal den. So to go back to our 2-3 studies, so the first one at the individual level, where we're going to look at the outcome of infection and the fitness cost from CDV infection. So this was published in a functional ecology. And so we're first going to ask ourselves, what drives the outcome of individual infection? So the outcome of infection is the results of 2 processes: the exposure, so the degree of contact between the host and the pathogen, and then the susceptibility, so the ability, the risk of infection given the contact, which of course depends on the ability of individual to allocate resources to immune processes. And so most of the studies focused on either the first part, so the exposure process, or the susceptibility process. And we were more interested in assessing the relative contribution of each process and how it can be mediated by social status of individual. And of course, the last part is the fitness cost in terms of survival and reproduction. So now, how does these processes how socially can modulate these processes? So as I said, as we said, we have a high-ranking and low-ranking female in the population. And so, of course, the exposure is going to be much higher among the high-ranking female, who tend to interact with more individuals to maintain the dominance status. And so the high-ranking female have higher exposure to pathogen. On the other side, those high-ranking female, they also have higher access to resources and better body condition, which reduce their susceptibility to contract the virus. So we tested different, we predicted what would be the outcome of infection under different hypotheses. So under the exposure hypothesis, if the exposure part of the process is more important, prevails over the susceptibility, we expect that high-ranking female have higher infection, that are more infected. And in that case, because they are more able to cope against the disease as they are have a better body condition, we expect fewer difference in survival between susceptible individual and infected individual. So no fitness cost of infection, no big fitness cost of infection. While under the susceptible hypothesis, so if the allocation to immune process is more important than the exposure, then we expect that low-ranking female being more infected and have a strong cost on survival, especially in low-ranking female. So what we did is we developed a multi-event capture-recapture model with uncertainty on disease states. So how does this work? So first, we're going to describe the biological process as the true transition between the different epidemiological states. So let's say that this individual started its life as healthy, susceptible individual, and then is going to survive with a probability phi and the probability of staying healthy, so not being infected by the virus, and is going to stay like this for 2 years, let's say. And then on the 3rd year, it's going to become infected given with a probability beta, given that it has survived. And then it has to, we have the transition to the immune state, given that it has survived between the in the next year. Let's say that it's then also survived in the last year and remain recovered. So this is the biological process, and now we're going to tease apart this biological process with the observation process, and we can define the likelihood of having a sequence of observation of states across the years, where we can decompose between two processes, the detection process and the assignment process. So, for instance, the probability of detecting a susceptible individual in the 1st year is the probability of detecting it given that it was healthy, and the probability of assigning it given that it was detected. And then the probability of detecting it in the 2nd year given that it was healthy, but the probability and the probability of not assigning it given that it was detected. And then the 3rd year, we can also have the probability of not detecting at all this individual, given that it was still healthy. And then the following year, the probability of not assigning this individual, given that it was detected and in the infected states, and the probability of having detected the individual as recovered, given that it was immune, and finally, let's say that we can also have the probability of not detecting this individual despite the fact that it was still alive. So basically, the likelihood of such model is going to be a big product of 3 big matrices: the initial states of the individual, the transition states, the transition matrix between states of epidemiological states, and the observation matrix, and the assignment matrix, the assigning correctly the disease states of individual. And then we multiply this across all individual, and we can infer some eco-epidemiological parameter. So to look at the results of infection and survival probability, here are on the y-axis, you have the infection probability of individual depending on their rank and their demographic states. And so we find a higher infection probability among cubs, low-ranking cubs, than in high-ranking cubs, so which support this hypothesis, the susceptibility hypothesis. While in the adults, we found a higher infection probability in adult high-ranking cubs, supporting the exposure hypothesis. So basically, this is also not surprising, because cubs have really highly depends on milk and resources, and low-ranking individual cubs have really struggled more in allocating resource in immune process. So all that is related to immunity is more important for the cubs and is going to affect more low-ranking ones, while adults have really high-ranking adults have really high contact rates and are going to be highly infected. Now, this is probably a shortcut, so I'm going to be very careful. But if we define basically virulence or as the death that is caused by the proportion of death caused by the virus, we can look at the difference between the survival of susceptible, healthy individual and with the survival of infected, and looking at how it, between the social status, how it can change. And as I said in the prediction, under the exposure, we expect that under the exposure hypothesis, there will be no big difference between survival of infected and susceptible individual, as it's more the exposure that matters, and the high-ranking female have really better susceptibility to shred the virus. Whereas, and so we found, and whereas for the cubs, we find a quite high difference for at least low-ranking cubs between survival of susceptible and infected. So just to summarize, we found that regarding the heterogeneity in the outcome of infection, high-ranking was more driven by exposure for high-ranking adults and susceptibility for low-ranking cubs, and which leads to quite high impact in terms of high impact on fitness costs for cubs. But in general, this is going to be offset and provide higher, not strong impact on the survival of adults. And if we want to look at how it's going to, of course, to really look at virulence, and if the strain is virulent in terms of on the population level, we need to now transfer and look at what's going to be the consequence in terms of population growth. So this is going to be the second part of my talk, and on how this how social traits can affect all these processes and in return the population growth. But also we looked at how it can also influence the basic reproductive number. So basic reproductive number is a quite important epidemiological indicator. So as you may know, it corresponds to the number of newly infected cases in a fully susceptible population that is caused by one single infected individual during its infectious period. So this is also quite important when we want to look at the long-term demographic impact of an epidemic. And but most of those studies looked at. Well, I will talk about it later. So this is the second part of the talk, and to determine what is the impact of an epidemic on a population level, and how the hyena population what's going to be its resilience and resistance to these CDV outbreak. So this was published in Communication Biology. Sorry. So what we did basically is to take again all the outcome and the results from the multi-event capture marker capture model. So just to summarize again quickly, because I was maybe a little bit fast with the susceptibility exposure hypothesis. What we got from this model is we find an effect of social states in the breeding transition probability. So the probability of becoming from non-breeder to breeder. We found also an effect of social states of so low-ranking, high-ranking in the transition. So it was basically it was more difficult for female to maintain high-ranking female to maintain their high-ranking rank to stay within the in the high-ranking part of the hierarchy. And then we found also interaction effects of demographic and social states on the infection probability and on the survival probability. So we use all these yearly estimates during 3 different periods: the pre-epidemic period, the epidemic period, so the 2 years of CDV epidemics, and the post-epidemic period. And all those demographic parameters allowed us to parameterize a big stage structured SIR matrix that is structured in 8 different sub-matrix, accounting for transition states between demographic states, transition states between epidemiological states, and between social states. And then we conducted asymptotic analysis to look at the host pathogen dynamics. So where we calculated asymptotic population growth rate for the host dynamic and this reproductive number, basic reproductive number, to capture the pathogen dynamics. So to go back to, so yeah, the basic reproductive number is a result of the degree of contact between individual and the pathogen, is the result of the ability of contracting and transmit the pathogen once exposed, and also the infectious period, the time of during which an individual stay contagious. And most of the studies that have looked at this basic reproductive number are sometimes not studied in age-structured population, and also not considering any change in the population growth. So in this matrix model, it was also mostly for time continuous model. And here we just developed a methodology to calculate the basic reproductive number in discrete time model, which was again already developed by Oli in 2006, and extended for so this highly aged and socially structured population, with accounting for the change in population size. So by accounting basically for growth rate, population growth in this calculation of basic reproductive number, we can have an idea of the change in terms of the incidence rates, the change in the spread of the disease, while accounting for the change in the population size of the host. And so this matrix model, then we applied and project this on population on a long-term time frame. And to get the asymptotic analysis, we could then get looked at the population growth rate of the host before and during the epidemic and after. So as you can see, the growth rate goes below 1. So you have a decline during the epidemics, and with a recovery, but not fully stable, because we're not yet at 1. And then in terms of the disease dynamics, we found, so yeah, sorry, my colleague just reminded me today, it was 569. The basic reproductive number of 5.69, which is similar to measles. And yes, and so here we found, we looked at the sensitivity. So we ran sensitivity analyses to look at how the variation, how when we change a demographic parameter of the host, how it's gonna impact in terms of the pathogen transmission. So the sensitivity of the basic reproductive number was really highly sensitive to the transition to the breeding states for high-ranking female. So in other words, it means that high-ranking breeders, female, is really the social state that matters the most in the population dynamics of the host. I didn't show it, but also in terms of the transmission of the pathogen. Yeah. And finally, we ran this matrix model under 2 different scenarios. One where we have the socially structured population with the low and high ranking transition, and another one where we considered no social structure in the population. So basically, we assumed that all the parameter, the ranking, the transition probability between ranks were the same, and that there was no difference in low-ranking, high-ranking survival in vitals rates, and then etc. And we can see that, so this is the one without social structure, and this is the one with the normal, capturing the normal ecology of the species. So you can see that, of course, we have the impact here of the CDV during the 2 years of the epidemic, and then with a slow recovery, but even slower, because it's about up to here, when we're not accounting for the social structure, which again means that really, which confirmed the fact that social structure in this population is contributing to the population, to the resilience of the population, and mostly through these high-ranking, the role of high-ranking females. But as I said, in this matrix model, we assume a constant parameter for between clans. So we didn't look at the group, the dynamics between clans. And now we ended up with a last question about how this is going to behave, the disease dynamics, and in terms of when we looked at the clan-structured population, so the dynamic between different clans, that in individual that lives in communal dens. So as I said, in the female have raised their cubs in and shelter their cubs in communal den. So during at least the first year of life, the cubs are not in interaction, are not in contact with members of other groups. And how this structured of communal den or communal nursery or kindergarten, I don't know how you want to call it, can affect the disease dynamics. And we looked at herd immunity. So this is a concept that I have not introduced yet. So the herd immunity in epidemiology, you probably also know, is basically the proportion and amount of immune individual we need in a population in order to the disease to either fade out or at least stabilize. So this was published in a Journal of Animal Ecology, and we developed an agent-based model in a SIR framework for species that have this particularity of living in communal nursery. And so just to summarize how it worked, we looked at two different scenarios. One scenario of a species that don't have those communal dens, where then we considered for those species homogeneous contact rate between between all individual of the groups, and another one where you have age-structured patterns in the between group interaction. So where only adult individual can be in contact with members of other groups, while all the other juveniles stays and are protected by this some kind of immunity barrier that is going to emerge from recovered adults. So this is our prediction, is that under this scenario, since there is really homogeneous group mixing, we're going to have always infected individual within the between groups, whereas in this structured, more age-structured patterns, there's going to be very quickly an immunity barrier that's going to emerge among the immune adults, and that are going to protect future pool of young cubs that are born. And we expect and predict that under this scenario, the epidemic will fade out more often and quicker. So, sorry. So this is what we found. Faster and stronger emergence of the immunity barrier under the communal nursery scenario, where you can see that during these are the times, how the disease spread through time. And this is the proportion of different type of group composition. And we have over the time, there's going to be at some point, the population is going to be composed of only groups that are sealed. So those groups of only recovered immune female or male and susceptible healthy individual. Whereas under the other scenario where everything is mixed, there's going to be the maintenance of open groups, and the disease is going to maintain. And so here is the the average probability of epidemic fade out over many, we consider also different scenario for many different life history traits of population. And we looked at, different heterogeneity, but this was just how the probability of epidemic fade out under the scenario where the kids are kept at home, so the communal nursery, and where the cubs are not staying at home. And we had, as a function of the reproductive, so this transmissibility of the virus, and we had systematically less fade out when there was not this age structured between group scenario. So finally, I don't know if I'm late. So this, to summarize, the summary, the conclusion of what we found in those three different studies. In terms of the impacts of pathogen, we can say that there was really a low fitness cost in adults, and especially in breeding females, which is as I said, which is the states, the demographic states that contributes the most to population growth. So consequently, thanks to that, to this offset between mediated by social rank, the population described and exhibited a higher resistance to the outbreak, but we found a slow recovery, especially when we don't account for the social structure. And then regarding what's going on, what's the effect of social contact within and between group in terms of the transmission and persistence of pathogens? Well, within the groups, as I said, we found that this dominance hierarchy structured in the spotted hyena can offset the cost of the high exposure that is going on within the clans, and and regarding the between group contacts, it confers a strong herd immunity thanks to the communal dance structure. And finally, very quickly, there are many different other questions can arise from this fascinating topic and model, where we could also look at the multi-host dynamics, because as I said, CDV, canine distemper virus, is a multi-host virus. So obviously, the interaction with other carnivores and the competition, inter-specific competition that is going on, for sure influence the full dynamics. And also how CDV, we have also different CDV strains have emerged in this population during the course of the monitoring. And we have more strains that are more prone to affect canids or those or felids, and how this can also affect the full dynamics and even evolution. So thanks for your attention. And I would like to, of course, acknowledge the grateful work and thank all my colleagues of our top hyena females, so Stephanie Kramer-Schat, Marian East, and Sarah Benhaiem, who led the project, and be very thankful to Heribert Hofer, Jean-Dominique Le Breton, and Jimenez, who also helped us in developing those studies.
Thank you, Lucile. And just to mention that Sarah is online also, so they will both be taking questions. So I think maybe we can start in the room and then in the audience online. So are there questions in the room?
Thank you very much for a lovely talk, Lucile. I was wondering, this is maybe not so much of a problem for you. So if it's not, then the question will be easily answered. But it's of interest for me for another project we have on a completely different disease. You know, sometimes when you have epidemiological data like you have, you're missing a fraction of individuals if the disease is resulting in high mortality very early on in the infection. So these would be individuals that have not been blood sampled or sampled and have died off very quickly after they contract the disease. I was wondering if this is an issue, because especially in your case, if this missing fraction is missing not at random, and that, for example, individuals of a certain social status will be more likely to die very quickly because of their body condition, for example, how would that affect your model? So I guess my question is it a problem for you? Is it a disease where you have these peaks of mortality very early on in infection? And if it is, did you manage to account for it or control for it? Thank you.
Thank you for this interesting question. I remember we asked ourselves this exact question during the development of the model, because indeed we miss, first of all, the cubs, when they are in the communal dens, they are really difficult to detect. So we don't, they're really under the burrows, and we don't see, So Sarah, maybe if you can answer, but I remember that's probably why we made this hypothesis that they're all susceptible, or do you have an idea?
Yeah, so yeah, thanks a lot. Just a quick thank you around Lucile for accepting to give the talk and presenting our project. I hope you can all hear me well, and thank you for the interesting question. Super great for the interesting question. So my first thought would be that first of all, the model is a bit general for this disease, because as Lucile said, we are looking at a yearly time interval. So we are considered that an individual that is infected for just for a couple of days or weeks will be in this infectious state for an entire year. So it's not an entirely perfect match in the terms of durations of the infectious period in reality and in the model. I think that for this specific disease, it's possible that we missed some cubs that would have died very shortly after the detection or the observation of clinical signs. What I think is probably so when cubs are around 3 months of age or 1 month of age, you start seeing them a bit more regularly, like when the mothers come to the communal dens and call them. So for nursing, so we see these cubs during this period, so it's possible to monitor the clinical signs during this period. It's a bit more of an issue maybe if the cubs would die very early during the first weeks, or we only see them shortly when they come out of the den. So it's a bit difficult to observe any clinical signs during this period, because as Lucile said, they spend most of the time in the communal den. And the sampling is also limited for cubs that are very, very young. We only get fecal samples when they start moving a bit around the den. So after 3-4 months of age, this is our earliest age. And if by chance we see a carcass of a young cub, but then it's also a bit difficult to access them. So I would say that maybe looking at patterns of mortality in terms of age, in terms of social status during these epidemic periods might give us a bit more information about if there are any biases that are linked to mortality. If some classes of individuals are more or less likely to die because of infection. So yeah, I think for this fine scale question, maybe individual analysis would be appropriate.
Yeah. Yeah, thanks, Lucia. I wonder if the level of inbreeding could also have an impact. I'm not sure this is relevant to your study system, but in a completely different system in social spiders, the group of Trine Bilde at Aarhus University just inferred that inbreeding could lead to depression in the immune response. I wonder whether this point could be relevant in your study system. Of course, in the spider system, the beauty was that they could compare social and asocial species. And the second question follows on whether overall, and this is what you started a bit to explore, comparing hyenas and zebras, whether there may be a similar other system with a different social structure which could allow you to explore these sort of things.
I'm sorry, I'm not sure if I got the second part. If in another species with a different social structure, it will also affect this inbreeding
yes.
cost. yes, good. Well, first, regarding on the cost for hyenas, well, for sure, this inbreeding issue of life in groups is the general cost of in terms of the impact for the impact of pathogens. And for hyena, I'm not sure if CIRAD, is there a high inbreeding rate within clans or?
So first of all, I'm not an expert on the link between the genetic background and immunocompetence, so I'm not sure I can say much about it. But generally, no, it's not expected that there is such a high level of inbreeding. As you probably know, the Serengeti ecosystem is quite a large area, and males migrate will disperse to another clan. So there is some exchange between these clans that are in different parts of the Serengeti ecosystem. But it's very, very possible. I think we neglected all this genetic background in the response to infection. So it's very well possible that these types of factors will determine whether an individual ends up becoming recovered or not becoming infected, and so on. So I think it's an entire part that we didn't consider here.
But yeah, I think for sure for different species, this impact of inbreeding is gonna be more or less modulated or buffered depending on the level of substructured there is in terms of interaction within groups. So if you take the wolves, for instance, there's this structured where there's only a alpha and an alpha mates, and to avoid inbreeding, and there is less, maybe more structured interaction between individuals that can also offset impact of pathogen transmission within a wolf pack, for instance.
Okay, well, thank you very much for your talk. I think it was amazing to have such quality of data where you can monitor the disease progression and transmission in a wildlife species. I've got a comment about Anne's question about the possibility of missing some individuals that would be dead before they emerge from the burrows. I was wondering, I assume with such a lifelong immunity, they must, mothers that are lactating must transmit protective antibodies to cubs. So possibly the cubs are protected up until they emerge, and it might be that you're not missing that many cubs in that period, but I don't know specifically about this disease. It's just a thought. And second, I had just curiosity question about the wider system and whether the epidemic that you described in 93, 94 was approximately the simultaneous with the epidemic that decimated the lions in the Serengeti, and then whether there was further epidemics boosts afterwards, after 93, 94, or what's the general dynamic? Is there any irregularity in these disease waves or in your system?
So yes, regarding, thanks for your question. Regarding the vertical transmission and the transmission of antibodies from maternal milk, from maternal to offspring. Sarah, can you remind me? So we basically made the simple assumption, at least in the matrix model, that there was no vertical transmission and no transfer of antibodies, that all the individuals were born susceptible, to simplify. But Sarah, do you want to add something on that?
So no, yeah, as you said, I think it's a very good point, and we had thought about this, but I think basically the fact that we consider the cubs as susceptible in their first year of life also is related to the fact that the mothers would, the recovered mothers would transfer antibodies through lactation. So yeah, I think cubs generally, if they are not exposed to the disease, are likely to be protected to some extent during this lactation period. So yeah, that's a likely process, yeah.
But I think it was just to some extent, no, regarding CDV. I
yes,
don't remember.
Yeah. Well, it's not been tested or looked at or studied, but it's the assumption that because antibodies were found in milk of other mammal, free-ranging population, it's likely that there will be they will confer some degree of protection against CDV. Yeah.
Then regarding the lions population, you know better, but yeah, it's the same. It has affected 30% of the lion population decline, whereas the hyena had less impact. So maybe you want to, you know better this difference between.
Yeah, so it's indeed the same famous virulent epidemic of CDV that occurred in 93-94. And as Lucile said, like 30% of the lion population was thought to decline, and including the adults. And the effect was particularly strong and in the lion population because of this impact on all age classes, whereas in hyenas, mostly the cubs were showing a decreased survival. But on the other hand, hyenas seem to have needed more time potentially than lions to recover, and probably because of this slow life history and long dependence on milk, the small litters that are produced. And there were apparently other cases of CDV infections after this major epidemic. So in I think it was 99 and 2000, they were called silent epidemics. And the main difference, according to research from colleagues in my group, is that there was a different strain during this main period. And the strain that occurred during the epidemic in 93-94, it was a highly virulent, because the non-candid species such as hyenas and lions were not adapted to this strain, and that's why the impact was particularly important. In contrast, the strains of CDV that occurred before and after were genetically different, which probably explained that they were silent epidemics.
Thanks. So we will now take a question online from Enrique Frago. Enrique, you can unmute your mic.
Yeah, can you hear me?
Yes.
Yeah. Hello, thank you very much. Enrique Frago from CVGP here in Montpellier. Thank you very much, Lucile. Yeah, I was just wondering, it seems that the disease is, the social life is quite advantageous to reduce this disease, but have you seen any costs associated to living in groups? For example, I don't know, keeping kids at home may reduce encounters with other individuals from other groups and exchange information or learning skills. That has been observed, for example, I guess, in other mammals. Just a curiosity about the costs of living in groups in these hyenas.
So you mean learning, transfer of information and learning abilities? Do you have an example, or I'm not sure I follow? Sorry. Hello.
Yeah, well,
Enrico.
I mean, my only example is with humans, maybe, that keeping the kids at home all the time, it can reduce learning skills, for example. And I guess that I'm sure that in mammals, there has to be examples where they exchange information between groups of animals, for example, on chimpanzees, for example. I'm sure they exchange skills about exploiting resources. I mean, I work with insects, so I cannot give any more precise example, but yeah, I just thought that maybe this isolation can imply.
Yeah, like to make a retarded young. I see.
Yeah.
No, well, for sure, those cubs at some point they follow after, once they have. At least in the hyena context, Sarah, you can correct me, but there's really a high investment in lactation and maternal care during this period where they stay within the dens. And then as soon as their body condition and everything is, as soon as they're ready, they're going to be following the mothers in the hunting trips. So I think the learning skills are there anyway.
Yeah, like you said, I think most of the learning skills in spotted hyenas are transmitted between mothers and offspring within the same clan, and other related family members that the young developing cub would experience. So all the contacts with all the closely related female, the aunts and the sisters and so on. So there is this passing of information within the clan and within the matriline. But most likely there is some inadvertent information that is transmitted between clans. I can imagine between regarding information about location of migratory prey, for instance. It's likely that that individuals or females of a clan will follow or use information about alien hyenas passing through their clans, about where the migratory prey are. So I can imagine that's this sort of information, but I'm not sure it's so there is potential contact between the adults of different clans, and this could already allow to have some transfer of information of this kind. I can assume, but I'm not sure that is detrimental for the developing cubs. I have not thought about it, but yeah, I think most of the learning is through the interactions between the mother and her offspring.
Okay, thank you very much.
You're welcome.
So actually, I had a question about your first study about the exposure hypothesis. And I was wondering if you took age into account, because I thought that maybe dominant females, they tend to be older and their immunity will change, not because, and okay, they're dominant, so they have more social contacts, but their immunity can also be different on average because they're older compared to low-ranking females. And so how does, like, did you take age into account in your model? And if you see that they are more infected, this dominant female, is it because of a higher exposure, or is it just because they are older?
Okay, so you mean the exact age and not the at which stage they are, like
yeah,
no, we didn't account for age. That's a good question. And how it could affect. Well, regarding the fact, maybe you correct me, Sarah, but I'm not quite sure that we can say that it is really correlated, the rank and the age, because hyenas are so complex. But as I said, the offspring irritate the social status of females. So just
yeah, it's not a social. So that would be true for males, for example. When they join a new clan, they are at the bottom of the hierarchy, and as they age, that tenure in the clan increases, and so the high-ranking males would be older. So there's a relationship between age and social status in males and females. If anything, that would be different, because the very old high-ranking females, so there tends to be a decline in the social status for the top-ranking female, as their daughters or other closely related family members would try to take their position, as it's seen in some primate species such as baboons. But that said, yeah, we didn't think about any potential effect of aging on immunity. So, but I don't think that it has an effect on any kind of bias in relation to the social states. But one thing maybe that is important for hyenas is that there is no menopause period. So the very old females, I think the oldest female that we have was 20. She died at 21 years of age, and they keep reproducing. So you see a decline in the reproductive success. The litter size tend to be smaller. They tend to show a lower offspring survival. But they still reproduce. So I'm not sure that whether this gives an indication about still a competent immune system. That's something we would like to look at in the future as well. But it's likely that it's just very in the oldest age, if anything, I would suspect.
Thank you. Maybe one last question? No?
Do we have time? Yeah, sure. I think it's related to previous question, but my question is that in the susceptibility hypothesis, the link between social rank and immunity is because you are high ranked, and so you have more resources, and so you are in a better shape. But what if the relationship is in the other sense? You are high ranked because you are in good shape for genetic reasons, or for because you are in the good family as it is inherited, or because of inbreeding, as it has been said, or because of maternal effect. Did you think about the possibility that the relationship is the reverse?
The real causality relationship. That's a good question. Yeah, good question. Thank you. So if I would tend to say that, okay, those interlinks between health, social states, and fitness is really complex. And as I said, it's difficult to really, despite using statistical model that can control for confounding variable, it's always very difficult to really quantify and examine the causality. But I would like in our capture mark recapture model, since we used those also the transition between low ranking, high ranking states. So there was also this parameter, this demographic, this social parameter of the probability of switching from one state to another within the rank. It also somehow account, somehow also account for the full dynamics in later the infection probability in terms of stage, social state specific. I don't know, Sarah, do you want to?
Yeah, I'm not sure I entirely understood the question, but so just going to reformulate it. Are you suggesting that actually high-ranking females in the susceptibility hypothesis do not have reasons to be less infectious or becoming less infected than the low-ranking females? Because so we based this hypothesis and these predictions based on research that we had in relation to disease and social status, as found in other species. High-ranking females, because they have this better body condition and higher reproductive success, are expected to be less infectious and become less infected with some parasites or some other viruses. So that's been found in previous studies, also in other study populations. And if we look at the effect of rank on parasite infection, you also see this relationship that the high-ranking female are less infected with parasites. So that was the basis, basically, of this infection. So we think that high-ranking females, because of their higher access to the benefits of having priority of access to food inside the territories, will likely have more opportunities to allocate investment, to allocate energy into immunity, which seems to be shown by previous studies. So I think that, yeah, that was the basis of the susceptibility hypothesis, but I'm not sure if it was your question exactly.
Well, my question was really on susceptibility, and just I thought if you have inbred and non-inbred cubs, non-inbred could more likely become high-ranked, and inbred not, and inbred would be more susceptible than non-inbred, to take an example.
Yeah.
But it can be maternal effect, it can be a non-genetic effect, just bad luck. So that you have some individuals that are interestingly more susceptible because their physiology is not good, you know, and they cannot become a dominant female.
No, so we don't think there's this kind of traits or traits related to health or body condition or fighting ability will influence whether a female become high-ranking or low-ranking. It's really this silver spoon effect that the high-ranking female supports her offspring during the interactions with other clan members. As a result, her own offspring will then become high-ranking as well, and then same process for the females at the bottom have of the hierarchy. And as Lucile said, in hyenas you see that size doesn't play a role in determining the outcome of interactions with small cubs learning that they can dominate adult males, or injured hyenas still. So it's probably going to be, to some extent, if a hyena is very injured, it's possible that this kind of affects her social status, that she will decline in rank. But it's more like a cognitive and learning process. It's related to this in the maternal inheritance, and then later on, when you're adults, the number of coalition partners and your social support that you get.
Okay, thank you.
Thank you, everyone. And thank you again, Lucile and Sarah, for this very interesting presentation.
Thank you. And thanks, Lucile, again.
So see you next week for the same on Friday next week.
Bye-bye.
Bye.